Okay, in today's lecture, we're going to be doing a quick review of photosynthesis, as well as talking about alternatives to just the regular old vanilla photosynthesis that plants can do. So remember, this is called the light-dependent reactions. And in the light-dependent reactions, we are taking a light energy, and we are using it to make these high-energy molecules, this NADPH and this ATP. When light comes in, remember it's going to excite this electron, and in order to replace that electron, we kind of need to split water and steal an electron from there. When we split water, what happens is oxygen is released. But why are we making these high energy molecules in the first place? Well, the answer to that question is we're doing it to fuel the light independent reactions. We're doing it to fuel the process of actually taking carbon dioxide out of the air and making it into sugar. So here is what the regular old-fashioned Calvin cycle looks like. You have CO2 coming in, rubisco is fixing that to RUBP. Some other steps are happening during the process, which involve those high energy molecules from before, and ultimately we are creating sugar. We regenerate RUBP, we regenerate rubisco, and the process goes on. But the problem is this molecule, rubisco, the most common enzyme in the world, kind of sucks at its job, just plainly put. Rubisco is not super good at what it does. All rubisco is supposed to do is take carbon, fix it to RUBP so that we can use it and turn it into sugar. All rubisco is supposed to do is fix carbon. But rubisco has this problem where sometimes, fairly often actually, it will fix oxygen instead of fixing carbon. Now, whenever rubisco does this, it's kind of bad for the plant. It's super energy inefficient. It costs the plant basically energy. Rubisco is not really the best at its job. So if you're a plant that is in a hot and dry environment, your stomata are going to close. So when things are either hot or they're dry, your stomata close up. And that should maybe make some sense to you, because the job of that stomata is not only to allow the plant to exchange gases with the environment, it's also to help the plant from losing water. Because if you're a plant and you're just opened up, if all those stomata are just open to the environment all the time, you're going to get dried out super, super quickly. And so in any plant, whenever things are hot or whenever things are dry, those stomata close. Now, what are we generating in the light-dependent reactions? We're generating high-energy molecules, but we're also generating a whole bunch of oxygen, right? Remember, we had to split that water molecule to regenerate that electron. So if you're in an environment where your stomata are closed, oxygen is going to build up. And when oxygen builds up, it just makes it so that it's much more likely that rubisco is going to have this problem, because it has a whole bunch more oxygen around, right? If you were inside of a cell, a plant cell, where there was no oxygen, rubisco would never be able to photorespirate, right? Rubisco would never be able to fix oxygen. It would have to fix carbon dioxide. But if you build up oxygen more and more and more, your cell is going to run into that problem more often, because those stomata are closed, gases are not able to be exchanged. So one of the ways through which plants that live in hot, arid environments can deal with this buildup of oxygen is what's called crassulacean acid metabolism photosynthesis. Not a term, you should know CAM, but you don't need to know crassulacean acid. But in CAM photosynthesis, basically what plants are doing is they're storing carbon dioxide. So here during the night, they can bring in carbon dioxide and store it as other things besides just pure CO2. And then they can undergo the light independent reactions during the daytime. So in CAM photosynthesis, they're using a temporal trick. They're changing when they're doing the steps of photosynthesis. In C4 photosynthesis, they're actually moving where rubisco is elsewhere to these specialized cells called bundle sheath cells. So in these bundle sheath cells, your concentration of oxygen is not going to be super high, even if you have to have your stomata closed. So in C4 photosynthesis, you move the location of photosynthesis. In CAM photosynthesis, you change the timing. Yeah, I think that's about all I had to say about these different kinds of photosynthesis. So remember, try to understand how the logic of this works. So if you are in a hot environment, you have to keep your stomata closed, but you're going to be building up oxygen. If you're building up oxygen, rubisco, which again kind of sucks, is going to accidentally photorespirate, which is going to cost you some energy. So in order to deal with that, if you live in a hot spot, you're going to either change when you photosynthesize, or change when you're undergoing some of these processes, or you're going to change where you're undergoing some of these processes. Okay, and just so you guys are, so we're crystal clear, these are sort of two different kinds of plants. You're not going to have CAM photosynthesis and C4 happening in the same plant. For a further discussion of this, I would recommend checking out Khan Academy, and I'll actually leave a link at the bottom of this page so that you can easily go find that. But they do a pretty good discussion of it over there as well.